The uncondensed chromatin or the euchromatin, it resembles beads on a string when viewed under the electron microscope. So in the interphase, when the cell is not dividing, the chromosomes are not present that time. So all the chromatin that is in uncondensed form. So in that form, if we see the chromatin under electron microscope, it appears beads on a string. Each bead is a nucleoprotein complex, which is nucleosome. As you can see in this electron micrograph, there are so many beads on the string like this. So these beads are called nucleosomes. Actually, these are nucleosomes which are present. Each nucleosome is formed by winding DNA fiber around a protein assembly, and this protein assembly, it consists of 8 histone molecules, and then the two nucleosomes in this string, the two nucleosomes are connected through the DNA, which is called linker DNA. Here in this you can see two nucleosomes. One is this and the other one is this, and each nucleosome consists of 8 histone molecules, and the two nucleosomes are connected through this linker DNA. So this is the structure of uncondensed chromatin. The size of the linker DNA between the nucleosomes is variable among different organisms. Even this length is variable in the same organism between different organs or tissues. For example, if the chromatin is isolated from one tissue, the length of this linker is different from the chromatin isolated from some other tissue. So this length is variable between different organs or tissues of the same organism and between different organisms. So how can we study the length of this linker DNA? This linker DNA can be digested with nucleases. So the nuclease, after nuclease digestion, we can find out the length of this linker DNA. The length of DNA that is wrapped around nucleosome, that is wrapped around histones in a nucleosome, it also varies from one organism to the other organism. The length of the whole DNA that is wrapped around 8 molecules of histone, this length varies from about 170 to 240 base pairs. So what is the variation of the length of linker DNA and the length of this wrapped DNA? The significance of this variation is still unclear. However, if we continue the digestion of chromatin, the prolonged nuclease digestion of the chromatin cleaves additional nucleotides. So after slight digestion, a nucleosome contains some extended DNA molecule, but after a prolonged nuclease digestion, these extended DNA is also digested, and then there remains a single core, which is called nucleosome core particle. So this nucleosome core particle, which remains after prolonged digestion, it consists of an octameric protein complex, and this octameric protein complex contains 8 histone molecules. 2 copies of each H2A, H2B, H3 and H4. So all these 8 histone molecules and about 146 base pair wrapped DNA fragment. So this constitutes this nucleosome core particle. Here you can see this is the chromatin fiber, and if we digest this fiber with the nuclease, the linker DNA, this will be digested, and these nucleosomes will be produced. And if we keep on digesting these chromatin fibers with the nucleases, then it results into nucleosome core particle. Here you can see this is nucleosome core particle, and in this particle there are 8 histone molecules, and around this octameric complex there are hardly two rounds of this DNA molecule. So this is nucleosome core particle.